Practical work number 1. Structural and functional organization of the sensory system for the formation of dental pain. The trigeminal nerve is a sensory nerve which innervates this region. At the peripheral nociceptors in the orofacial region, after receiving repetitive noxious stimuli or an excessive uncontrollable inflammation, the first-order neurons in the trigeminal nerve will develop increased pain signals that are projected to the trigeminal ganglia. After that, the pain signals are sent to the second-order neurons in the trigeminal nucleus caudalis located in the brainstem. Then the signals will be further projected to the third-order neurons in the thalamus via the ventral trigeminal thalamic tract. Therefore, numerous studies supported that the physiology of orofacial pain is related to the trigeminal nerve and trigeminal pathway, leading to altered pain perception in the relevant area of the somatosensory cortex and associated somatosensory cortex. Practical work number 2. Features of forming saliva while affecting rejected substances on the mucous membrane of oral cavity. For the work we need distilled water, 5% solid of glucose, lemon juice, a solution of bitterness or salt, 3 tubes with divisions, a tripod. 1 milliliter of water is injected into the oral cavity, then spit it out into the tray. After rinsing, the saliva is collected into the tube during 2 minutes. Then repeat the experiment with the 1 milliliter of 5% glucose solution, 1 milliliter of lemon juice, and 1 milliliter of solution of bitterness in 4 different tubes. After that, the volume of saliva in each tube is compared. Data is entered into the table. Different volume of saliva is the result of conditioned reflex secretion to various stimuli.
Practical work number 3. Determination of thresholds of pain sensitivity to electric current, electroaltesimetry. We need electrostimulator ESL2, intermediary transformer, indifferent and active electrode, cotton, spirit, physiological cell solution. The research is conducted on a person. An indifferent electrode of a large area is strengthened on the palm of the right hand. Irritation frequency 5 pulses S, pulse duration 3 ms. The ECL2 electrostimulator is connected to the irritating electrode through a separating transformer. The irritating monopolar electrode is alternately placed: A. In the chin area, the exit point of the chin nerve on the left and right. B. In the area of the mucous cheek and the level of the eye tooth, left and right. C. In the area of the central upper and lower gingival papillae, gradually increasing the intensity of irritation. They bring it until the subject has a clear sensation of painful injections. Measurements at each point are carried out 3 times. Data is entered into a table. The most sensitive areas turned out to be the upper lip and the region of the right gingival papillae, which is the result of asymmetry of innervation. Practical work number 4. Electroodontometry. The research is conducted on students. An indifferent electrode is connected to the plus terminal of the device, placed on the palm of the subject and fixed with a bandage. The end of the active electrode is wrapped with a thin layer of cotton wool, moistened with water, and applied to the sensitive point of the tooth. Thoroughly dry the test tooth with tampons, lay cotton rolls. Data is entered into a table. The condition of the examined teeth, according to the data of electroodontometry, varies in different subjects but is within the normal range.